Well, a warm welcome to this series of video presentations on the role of bisphosphonates in the treatment of Paget's disease of bone. So my name is Stuart Ralston, I'm chairman of the Paget's Association, and we're presenting these videos in celebration of Paget's Awareness Day on the 11th of January 2021, which marks the 207th anniversary of the birth of Sir James Paget. So it's my pleasure today to welcome Professor Graham Russell, who has very kindly agreed to talk to us about some of the early research he conducted on the effects of pyrophosphate and bisphosphonates in bone metabolism. Welcome, Graham.
Hello, Stuart.
Hi. Well, we know each other very well, but maybe you could say a few words about yourself and your background in the field for the few people that might not be so familiar with you.
That's a nice introduction. Well, the story of how bisphosphonates came about is kind of interesting because it starts, as some things do in life in scientific research, completely by accident and serendipitously and so on. So very briefly, I was a biochemistry student at Cambridge, and when I finished my undergraduate, I decided to do a PhD in Leeds in a medical research council unit and to study kidney stones, which was the topic that they were working on. And to cut a long story short, the question of how calcium kidney stones were formed wasn't fully understood. And there were a publication appeared very early in my PhD from Herbie Fleisch and Bill Newman in the US, suggesting that the body had a natural water softener in it. And it turned out later that this was pyrophosphate. And pyrophosphate was obviously a very good candidate for having something to do with kidney stones. So I got involved in that as a PhD project and developed assays and so on. And this led me to make contact with Herbert Fleisch, who by that time had returned from the US to Switzerland in Davos. And I visited there to spruce up the methods, and eventually, very quickly in the early 60s, ended up working there. And the effort in those days was really to look for mechanisms of inhibiting calcification, kidney stones being an example, but also blood vessel calcification and so on. And we worked on pyrophosphate, but the problem was that pyrophosphate couldn't easily be given to animals or people in particular and see any results because it was broken down by this enzyme alkaline phosphatase that is present in the body. And alkaline phosphatase is the same enzyme as one measures to measure how active your Paget's disease is. So we looked around for chemically stable analogues of pyrophosphate, and that's what we found. They were out there, the bisphosphonates, they'd been made a few years before, and the folk who were most interested in them were actually at Procter & Gamble in the US in Cincinnati, who were studying these sort of compounds as potential additives to detergents and to toothpaste to prevent calculus deposition and so on. And we started working on that and showed that they were indeed stable. They inhibited calcification very nicely, even when given by mouth. But the key step was that they also blocked the dissolution, the sort of breakdown of calcium phosphate crystals. And that led us to think sort of sideways that, well, maybe they would do something to bone resorption. So we did the appropriate experiments, and that's what we found, and that sort of led on to looking at diseases of bone resorption in humans, first of which was Paget's disease. So that's the brief history of that bit.
Thank you very much. It's fascinating, and the connection with Procter and Gamble is fascinating. You know, these compounds that they were using to soften the water and DAS and such like ended up being studied in the bone research laboratories. Now, if I can take you back to, I know you published with Herbie on pyrophosphate, but also on the bisphosphonates, your paper in Nature in 1969, looking at clodronate, which is still around these days, in a model of immobilization. And that showed pretty dramatic effects in preventing immobilization-induced bone loss. That must have been a very exciting result when you got that. And Yeah. So what did you think then? And is that why you decided, gosh, this might be interesting to study in humans, basically?
Yes, absolutely, because demonstrating that you had a very effective inhibitor that worked also by oral administration, um, against bone loss, meant that you could look at the whole spectrum of bone diseases where bone loss was a feature. And of course, Paget's was the most obvious of those. And that's really why the first studies were done in Paget's disease. But as you know, bisphosphonates are used these days in osteoporosis and in other settings. In fact, they become the leading drugs for a whole variety of disorders of bone resorption.
Yeah, amazing. And I've
And
used
there are
some.
about a dozen, as you know, there are about a dozen compounds that have been developed over the years, and they've gradually got more and more potent and selective and effective.
Fantastic. And at that time, because you were working, I think, on the hypothesis that the compounds would incorporate into the mineral and stop dissolution, I think you actually mentioned that. Was that what you thought was the mechanism of action? And then obviously we know different now, but maybe you could just take us through that.
Yes, that's in itself an interesting point, because we thought it was a physicochemical effect, just an effect on the release of calcium from bone that was being blocked. And we very quickly began to realize that that couldn't be the whole explanation, and that there must be effects on the bone resorbing cells, the so-called osteoclasts. And it took ages and ages to nail down the mechanism of that. And it wasn't until nearly 20 years later that we managed to work out what that mechanism was, with Mike Rogers and other people. Actually, in Sheffield, we did those studies. And it's a good illustration that you don't always have to know ahead of time how a drug exactly works for it to be useful. It's a bit like the aspirin story, where it took, ages before people really knew that mechanism too.
Okay. And you obviously finish your stint with Herbie, maybe the late 1960s, early 1970s, because you then appeared in a paper actually using etidronate. And so did you move back to Oxford at that time, like to do this study, or what happened then?
Yeah, so what happened was I spent 2 or 3 years in Davos, and came back to the UK, to Oxford actually, to finish my clinical studies in medicine. And it was actually during that time that I linked up with Roger Smith, who I think you're going to be talking to. And he provided access to the patients, and we worked together, as it were, on the first studies in people with Paget's disease. And as you say, that was published in 1971, which was a very short time after recognizing that these drugs could be potentially useful therapies.
Absolutely fantastic, Graham. From water softeners to a class of drugs that have revolutionized the treatment of Paget's and many other bone diseases. Now, just before we close, I gather that congratulations are in order, because I heard through the grapevine you'd recently been honoured by the award of the Sir James Black Award from the British Pharmaceutical Society for your work in this area. So fantastic. I guess you were well familiar with the work of Sir James Black, but that must be a great feeling to have been given that.
Indeed, that's a very pleasing award to be given, because it's essentially for drug discovery. It's the British Pharmacological Society, and they honour Sir James Black, who actually worked on drugs that will probably be very familiar to many people. He developed the beta blockers for heart rhythm, like propranolol, and then the drugs that affect acid production in the stomach, like cimetidine. And he ended up getting a Nobel Prize, as well as becoming, I forget the exact role, I think it was in Dundee, wasn't it? He was principal of Dundee University. A good Scot, Stuart.
Well, thanks very much, Graham. Well, I'm sure everyone watching these video diaries will agree that this is it was a very richly deserved award. And thanks so much for sparing the time to talk to us today. As you mentioned, we're hoping to speak to Roger Smith in a later interview in this series, but it's just been so brilliant to hear your story of the very start of the bisphosphonates, and hopefully in the video series we'll track them through their development in Paget's disease as time has gone on, and we've developed, and more potent compounds have been developed. So thanks once again.
My pleasure. Thank you, Stuart.